Hello everybody and welcome to yet another video lecture. So the topic is on G protein coupled receptors. So we will look into the receptor, its structure, the way it receives the signal and the G proteins that get associated or dissociated from the receptors and how they pass the signal from the receptor to the inside molecules that act as messengers, that is the secondary messengers. And so this is what we are gonna cover in this video lecture. So the G protein coupled receptors, they are also called as the serpentine receptors or 7 transmembrane receptors. That is serpentine receptors or 7 transmembrane receptors. Why? Serpentine is kind of a snake-like, right? So when we look, it is kind of a snake-like. See this. So it is called serpentine, and 7 transmembrane receptors because you can see over here there are 7 transmembrane proteins which are the basic component of our receptor. This is 1, 2, 3, 4, 5, 6 and 7. So we have 7 transmembrane proteins which are the essential body or the body build of the receptor. So hence the name. Now let's take a better look into the whole structure of the receptor. So when we look into it, we get to know that the receptor, it has an amino terminus, N-terminal side towards the outside of the cell and a COOH carboxy terminal towards the cytosolic side of the cell. And then there are connecting loops, these black ones, which connect the 7 transmembrane proteins with each other. So these are called connecting loops. The transmembrane proteins are essentially alpha helices, and this part of the receptor, this part will act as the ligand binding zone, this outer part, and the inner part it is gonna act as the binding site for intracellular signaling molecule, since it is the cytosolic part, right? And this thing that we see over here, G alpha, G beta and G gamma, these are the G proteins. So it is for the timing, it's found towards the cytosolic part, near to the this receptor area. So it is found in close association with the receptor for the timing, and we will see its dissociation, association, etc. in the coming slides. So these receptors are initially in a conformation which is called inactive conformation. So when we look into the inactive conformation, it is stabilized by the non-covalent interactions between specific residues in this transmembrane alpha helices. So there are some non-covalent interactions between the alpha helices. And when the signaling molecule, maybe this is a signaling molecule, and when this binds to this ligand binding site, the conformational change occurs in the receptor. That is, this interaction, this non-covalent interaction will get disturbed, and then it acquires the active conformation. So what happens? The ligand binds to the ligand binding zone, and the non-covalent interaction between these alpha helices get distracted or disturbed, and then the receptor turns on or it becomes active. How? That is rotation and shifts. Rotation and shifts occur in the transmembrane alpha helices. These alpha helix, they rotate, they shift and they form a active conformation. Okay, so this active conformation will cause some changes in the cytoplasmic area, which will lead to the propagation of the signal. Okay, so this essentially what happens is that the active conformation, this conformation will cause an increased affinity of the receptors to the G protein. The G protein's affinity to the receptor will increase. That is, the G alpha, this area will come towards the receptor. It gets the affinity, the liking of the receptor towards this G protein, it is going to get increased. And as a result of this increased affinity, the GTP molecule will bind to the G protein, G alpha side, and then what happens? And this is the way in which the signal get propagated. So we will look deeper into it, but for the timing, hope you understood these things. It is also called serpentine receptor, 7 transmembrane receptor. It have amino terminal outside and COOH carboxyl terminal towards the cytosolic part. The G protein, that group, it is a trimer. It is present in close affinity to the receptors, and when the receptors become active, its affinity towards the G protein will increase, and the G protein will also be able to bind to GTP molecule. Okay. Now let's talk about something which is called signal amplification. Signal amplification. So it simply means that a single receptor, that is the receptor that we saw here, single receptors may have many G proteins in and around it, right? So a single receptor can actually activate many G proteins which are around here, and this activation of many G proteins by a single receptor is called signal amplification. Hope you understood. That is, a single receptor can activate many G proteins. So this is called signal amplification. And then let's look into the G proteins. G proteins. So these G proteins, they are heterotrimeric. So as we saw in the earlier case, that is, there is one G, maybe G alpha. Then there is another one which is G beta. And then we have another one which is G gamma. Okay, G alpha, beta, and gamma. So what happens is that these, as you can see, they are of different shapes and colors, right? I have represented it so because they are hetero, because three are different. All the three molecules are different, and they are trimeric because they are 3 in number. So they are heterotrimer, and it was discovered and purified by a person called Martin Rodwell. And Alfred Gilman. Okay, Alfred Gilman and their team. So they were the people who discovered it, purified it, and characterized it. So these proteins they have the ability to bind to GTP or GDP. So I was talking about the G alpha getting binded to GTP, right? So when I mentioned it, when I was explaining it, I explained it like when the receptor get activated, when a ligand binds, it binds to GTP. So binding to GTP essentially indicates the activation of our receptor. So there are three subunits, as I have said, and this alpha subunit is the one which is gonna bind to this guanine nucleotides. GTP is guanine triphosphate and GDP is guanine diphosphate. So this this G alpha have the ability to bind to GTP or GDP. That is, and one more thing, there is something called as monomeric G protein. Okay, this is something different. So there is something which is called monomeric G protein, and this is none other than our RAS. Okay, so this is a bit of extra information. Maybe it will be helpful for you. Maybe it can come as a single sentence in an option, so you can pick up the correct option. Okay, and our G alpha and G gamma. So it's actually like you can make a small correction over here, that is make it G gamma and this beta. So this G gamma and G alpha, they are gonna get linked to the double helix, the lipid layer of the double helix by covalent bonds. Okay, covalent bonds. So covalent bonds are gonna get playing, and this G alpha and G gamma, they will be linked to the plasma membrane by covalent bonds. Hope you understood. Now we will discuss about the on stage of our GPCRs. On stage of G protein. Okay, so what happens essentially, one thing, that is GDP will be bound to G alpha when it is in off stage or when it is in inactive stage. So when the G protein is on, that means the GDP will be replaced by GTP. So GTP will come and bind to G alpha. First thing. Second thing, the G protein, that is G alpha, essentially G alpha. So okay, G alpha will lose its affinity to G beta and G gamma. Okay, so this heterotrimer, they are gonna get dissociated when the receptor, the ligand binds to receptor and the G protein gets activated. At that point, this heterotrimer will dissociate. So that's second point. Third point. So third point, we can write it like trimeric complex will dissociate. And the fourth point is that G alpha is free because it got dissociated from the GDP, the G beta and G gamma, right? So now it is free to activate the effectors. And now what are these effectors? These effectors are what we call as the second messengers. Right? So this is about the on stage of our G protein. On stage, one, GDP will get dissociated and GTP will get associated to G alpha subunit. Then G alpha will lose its affinity to G beta and G gamma. They get separated. That is, trimeric complex will dissociate, and G alpha is free to activate the effectors, and these effectors are essentially the second messengers. Now let's look into the intrinsic control of G protein. Intrinsic control. So what does this mean? That is, the G protein, the activation, deactivation, everything is controlled by the cell and its environment, the receptors, etc. Okay, fine, well and good. But can the G protein have an internal control over its activation? That's what the question is. Okay, so this G protein, they have the ability, they can turn themselves off. Off how? By hydrolysis of GTP to GDP and phosphate. So what happens is that they can hydrolyze. They can undergo hydrolysis process, and then when the GTP becomes GDP, that essentially means that it is getting inactivated, as I already stated, right? It is essentially, this hydrolysis is actually of very weak activity. It is not like you add the receptor ligand binding occurred and GTP got bound and immediately it is getting hydrolyzed. No, it is very slow activity. The hydrolysis will occur very slowly. So it is under control. It occurs very slowly, so that it if the G protein is active for a very long time, then naturally by the inherent property it will get slowly hydrolyzed and then it will slowly get switched off. So this is an inherent property. So this hydrolysis is actually causing a conformational change. So as I said, over here we discussed, right, the binding of GTP to the G alpha is followed by the dissociation of G beta gamma. So what happens when GTP is getting hydrolyzed to GDP? Its conformational change occurs such that the affinity to it have the decreased affinity to effector, that is the secondary messengers, decreased effector affinity but increased affinity to G beta gamma. So everything that occurred in this previous one over here will slowly get reversed, right? The secondary messengers are no longer being produced. The G beta gamma affinity will increase, that is the trimer formation will occur, and thus it will become inactive. Okay, so this is like it will form inactive trimeric conformation or G protein, right? Now this is how it get on and this is how it get off by intrinsic property. Now there are extrinsic properties which we will discuss, but before we discuss that, we will discuss something else, that is the types of, or we can say flavors of our heterotrimer, that is of G protein, right? Now first let us discuss about GS, and this is actually adenylyl cyclase, 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 right? And this GS will activate the secondary messenger adenyl cyclase. Then second one is GQ, and it usually activate PLC beta, that is the phosphatidylinositol, that kind of pathway. So it will activate that is it will lead to hydrolysis of phosphatidylin inositol bisphosphate, okay, phosphate. and so on. That this is a different pathway. This pathway will get progressed with the G protein coupled receptors. The third one is that GI. It is an inhibitor. It inhibit the inhibit the adenyl cyclase. Then we have something called G1213, and this is actually an inappropriate activation lead to excessive cell proliferation and active malignant. It's kind of a cancer transformation. When it is when it is activated in an inappropriate manner. So this is the culprit where any inappropriate activation can lead to the formation of cancer, tumor, the malignancy, the dangerous ones. Okay. So these are the 4 types of G proteins that you need to know for your exam. And then we have something called, which I was talking before, desensitization. It's not P, okay, it's desensitization. Right. So this is the process that blocks or stops the active receptor from activating more G proteins. So we saw that the receptor is surrounded by G proteins, and when the ligand binds to the receptor, the G protein get activated by binding itself to the GTP. So it is the stopping of this process, right? So first one is step 1. Cytoplasmic domain of GPCR is phosphorylated. That is, cytoplasmic domain of GPCRs get phosphorylated. So the G protein coupled receptors, that is GPCRs, the serine threonine protein kinase family will recognize. There is the family of protein kinases. Kinase means kinase will phosphorylate. They phosphorylate a group. So there is this serine threonine protein kinase, serine threonine protein kinase, which will phosphorylate, which it can recognize the active GPCRs, and then it will phosphorylate it and it will make it inactive. So this is the first way. What happens? The cytoplasmic domain of GPCRs get phosphorylated, and this phosphorylation will will inactivate it. Now step 2. That was the step 1 and step 2. Competitive binding of arrestin proteins to GPCRs. So there are these arrestin proteins. Arrestins. So they bind. They compete. There are 2 things, that is G proteins, G alpha plus these arrestins. So they are a kind of similar things. So they will compete for the active site on the receptors. Okay, we have this 1, 2, 3, 4, 5, 6, 7. This 7 receptor kind of thing to which our G alpha is actually bound. But when the arrestins are also released, because we are aiming to inhibit the receptor, right? So to inhibit the receptors, the arrestins are released. So when the arrestins are also released, they get into a competition. That is, they will fight for this location where the G alpha was initially binding to the receptor. So this arrestin gets bound to this place, and thus it will inactivate the GPCRs. So this is the second way. Okay. Now, regulators of G protein signaling. Now we will look into the regulators. Right. So the accelerate hydrolysis process of GTP by G alpha. This regulation is actually by the accelerated hydrolysis of GTP by G alpha, right? So this activated hydrolysis will cause the conversion of GTP to GDP as we discussed. So this activated hydrolysis will ultimately inactivate the GPCRs, the G protein coupled receptors. Okay. Now let's get an overview, and before that I would like to discuss something else too. So that is like see, we have our G alpha, G gamma and G beta, right? And as I said, they are in the cytosolic part. So something is in the cytosolic part essentially means that they are hydrophilic. Now where will I write that? Yeah, they are hydrophilic. When hydrophilic, when they are in heterotrimeric form, they are hydrophilic. But I told you guys that even in the on stage it will get lose its affinity to G beta and gamma, and the trimeric complex will dissociate. So when this dissociated, the G beta and G gamma, this thing turns hydrophobic. So what happens? Since it becomes hydrophobic, is it suitable to remain in the cytosolic part? No. Hydrophilic molecules are essentially not suited to remain in the cytosolic part. So it when dissociated from the trimeric form, it will go into the lipid layer, right? So when it is in the lipid layer, they are kind of protected. The lipid bilayer, we know that this tail part is hydrophobic and head part is hydrophilic. So initially they were bound to the head part lipid region by covalent bonds because they were in trimeric form and they are hydrophilic. But when they are dissociated, they become hydrophobic, and this hydrophobic moiety will enter the lipid bilayer for its protection. And this entered thing, they have some role in other pathways, other receptors. So that is there, but for the timing you just understand these things, okay? So we talked about GPCRs, the serpentine 7 transmembrane receptors, the inactive conformation, and how it becomes active. Its affinity to GTP will make it activated, right? Then signal amplification essentially means that a single receptor can activate many G proteins around it, and the G proteins, they are in heterotrimeric form and they are active when they are bound to GTP, and when they are bound to GDP, they are inactive. And when the GTP binding occurs, its affinity to the G beta and G gamma will decrease slowly, and the trimeric complex will dissociate, and the G alpha is free to activate the effectors, that is the secondary messengers. Then we discussed about how intrinsic control occurs, that is the self-control of the G alpha. The G alpha will hydrolyze the whole thing, that is GTP to GDP. It is very slow process. It is not like that a receptor is activating a G alpha and suddenly the alpha is doing hydrolysis. No, it doesn't occur like that. It is very slow. That is, if the G alpha is on for a very long time, it will hydrolyze itself and make itself inactive. That's it, okay? Now this conformational change will reverse everything that occurred over here. That is, it will decrease the effector affinity and increase the affinity to G alpha and G gamma. Then 4 types of G proteins, they are GS, GQ and GI, then G12, 13. Then we talked about desensitization, that is step 1. The cytoplasmic domain of GPCRs get phosphorylated by kinase enzyme, that is serine threonine kinase, and it actually inactivated. Then another thing is competition between arrestins. So then we discussed about the regulators, that is hydrolysis of GTP, that is how the regulation occurs. And then we also talked about the hydrophilic nature of our trimeric compound, and the beta gamma get hydrophobic when it get dissociated. So that's what our class is about. Hope you understood and got a clear idea about the G protein coupled receptors. And it is a very important topic for the examination. So revise it well. All the best, guys.